Hi everyone, welcome to MedGoto. In this video, we'll be talking about cell death. This is going to be a continuation of the USMLA series that we started. Before we continue, don't forget, if you are yet to subscribe to this channel, you're really missing a lot. Don't forget to subscribe, put on the notification bell so that each time we upload the video, you can get the notification. So having said that, let's move on to cell deaths. Couple of days ago, we spoke about cellular adaptation. We mentioned that if you put too much of stress, right? So if you put too much stress on a cell, right, the cell can adapt, right? So we spoke about that cellular adaptation. And we mentioned different ways that a cell can adapt, either by, we mentioned hypertrophy, we mentioned hyperplasia, we mentioned metaplasia, and so on and so forth, right? And then we mentioned that if the stress is too much, so if the stress is too much, right, that can lead to cellular injury, or if the cell cannot adapt anymore, right, any of them can lead to cellular injury. And then with this injury, right, if the injury is too much, it can lead to cell death, right? And I will mention that there are two main ways that the cell can die, right? The cell that can die by necrosis, right? Or they can die by apoptosis. So for the purpose of this lecture, we're going to start with talking about necrosis. We're going to start with necrosis. So necrosis is a death of cells that often involve the acute inflammatory cells. And then throughout this USMLE series, right, anytime we mention acute inflammatory cells, right, always think of neutrophil. It's one of our main acute inflammatory cells, right? And then one thing that we also remember about necrosis is always pathology, right? It's never going to be a physiology process, right? It's always pathology. As opposed to apoptosis, which we can have physiology importance of apoptosis, right? And then when we look at necrosis, it's divided into several types based on the gross appearance, right? So based on the gross appearance of the features, right, we divided necrosis into different parts, right? So the first one, which we want to talk about, focus on this lecture, is going to be coagulative necrosis. And then what's coagulative necrosis? This is a type of necrosis in which the structural outline of the dead cells, right, are preserved. And I'm going to explain what I mean by that. So let's go to what I mean by that. In other words, let's talk about the mechanism. The mechanism of this type of necrosis, right? So commonly there are three main things, right, that could lead to coagulative necrosis. Although there are other things, but the three main ones that we need to focus on. Number one is hypoxia, either because of ischemia, right? And if you guys remember, what's ischemia? Ischemia is reduction, if there's decreased blood flow to the tissue, right? So if there's a decreased blood flow to the tissue, that's what we refer to it as ischemia. So either ischemia, right? Or if somebody is exposed to heavy metals, example like lead poison. And then the third one is ionizing radiation. So any of these three, right, can lead to coagulative necrosis. So with hypoxia, like we mentioned in our previous lecture, right, so if there's hypoxia, the cell will move from aerobic to anaerobic glycolysis. And with anaerobic glycolysis, all your glucose, right, will produce pyruvate. But because the pyruvate cannot go into the TCA cycle, into the Krebs cycle, the pyruvate is shunted to production of lactic acid, right? So, so if you have increased production of lactic acid inside the cell, that can decrease the pH inside the cell, right? So either of any of these three, right, either because of heavy metal, ionizing radiation, or because of accumulation of the lactic acid, any of these, right, they are going to denature protein. And that's the key word here, denature. They want to denature the protein. And in other words, inactivate the protein. Denature the protein. So the question is that what are the proteins? So any protein inside the cell, they are capable of denaturing those proteins. So for instance, they can denature the structural protein. And a structural protein, these are the guys that are responsible for the structure of how the cell looks like, right? So as a result of that, we want to end up with dead cells, right? So all those structural protein will be dead, right? And at the same time, they also inactivate enzymes, right? For example, like proteases. Right? So the proteases that are supposed to break down this dead cell cannot break down the dead cell. So what you end up with is just like you have a cell, right? We have the organelle inside, right? But everything inside the cell, right? They are dead. So they are dead tissue. So the structural protein inside the cell, they are dead. And the protease cannot break them down because either from lactic acid or the heavy metal producing free radicals or the ionizing radiation producing free radicals, they denature the enzyme. So as a result of that, the protease will not be able to break down the dead tissue. So when you look at the tissue, the structural architecture, right, or the structural outline of that cell is preserved. And that's the key word in coagulative necrosis. So that's why they call it coagulative, like a coagulation of dead cells, right? So that's where I imagine it like coagulation of dead cells. And that's where that word comes from, coagulative necrosis, right? So what are the microscopic findings that I'm expecting, microscopic findings that we're expecting in coagulative necrosis, right? So number one, like we mentioned, we expect to see a preserved cellular architecture. Number two, right, when you look at the nuclei, it's going to disappear. So when you look inside those tissue, we are not going to see any nuclei there, or they might be fading away, right? Whenever you use the word undergoing pyrolysis, right? And also, when you look at the cytoplasm of those dead tissue, right, they want to appear red or pink with eosin stain. So when you apply eosin stain, they want to appear red or pink, right? And sometimes you can see some textbooks will say eosinophilia. And so those are what we see with coagulative necrosis. And of course, the question now is that how are we going to get rid of this all these dead cells from our body? Thank God for neutrophil, right? So after 24 hours of all these dead cells, right, neutrophil will start to move in, right? So neutrophil will start to move in, and then neutrophil want to create inflammation, right? And by third day, by day 3, right, macrophage is going to come in, And the macrophages is going to remove those dead tissue, right? And it's going to remove all those debris, clear them out, and then granulation tissue can start coming in, right? Which is a process of repair. Right? And I hope this explained coagulative necrosis to us. In the next video, I'm going to use an example of myocardial infarction to explain the process of that coagulation so that we can see what happened in coagulative necrosis. And then one thing I want to mention before we conclude this aspect is, remember, anytime we have infarction, right? Infarction is basically the gross manifestation of ischemia. So if somebody has ischemia, the tissue can become infarcted. So that gross manifestation is what we refer to as infarction, right? So anytime we have infarction as a result of ischemia, right, to any tissue in our body, we normally get coagulative necrosis. The only exception that we need to remember is the brain, right? In the brain, we get liquefative necrosis, right? And this is very important for us to remember. They can ask us on the exam. So anytime there is infarction to any organ in our body or to any tissue, we get coagulative necrosis. The only exception is in the brain, whereby we have liquefative necrosis. And of course, we're also going to cover liquefative necrosis in this series. Thank you guys for watching.